and welcome to another episode of the Iraqi Nutrition Podcast. I'm your host, as always, Juma Iraqi. Before we start today's episode, I just want to mention that this podcast is available on YouTube, but you can also find it on SoundCloud, Stitcher, and iTunes as well. I know it has been a few weeks where I haven't been able to do an episode, but I'm glad that I'm back, and today I have a very special guest, Professor Don Layman. Professor Layman, thank you so much for agreeing to take the time and be here today to discuss a very interesting topic. We're going to talk about protein metabolism today. So thank you so much for joining me today.
My pleasure to join you. I look forward to talking about one of my favorite topics.
Me too. I think this will be a great episode. So for the listeners that might not be familiar with you, could you give us an introduction about yourself?
So I've been a professor of nutritional biochemistry at the University of Illinois for a little over 30 years. I actually retired from the university a couple of years ago, so I'm now an emeritus professor. My background is really protein and amino acid metabolism. I've looked at muscle development, both from malnutrition as well as athletic performance, but also related to things like maintaining body composition during weight loss for obesity and aging and diabetes and things like that. So I'm definitely muscle focused, sort of a muscle centric nutrition focus, and also sort of a protein centric nutrition focus.
Awesome. I think this is probably the episode I have wanted to do the most because it dives into the stuff that I'm really interested in, which is what's actually happening on a deeper level with protein and signaling. So I'm really looking forward to this episode. So to start off, we often talk about high protein diets, but do we have a definition for what a high protein diet actually is? Like, what's the amount of protein we're talking about?
Yeah, that's really a good question. And the answer is really we don't have a definition. It's kind of in the eyes of the beholder. I'll give you the short answer from my perspective, and then I'll give you a little longer answer. I think that a high protein diet is something above 1.8 grams per kg. And I'll give you a little background to that. Most people, when they hear the word high, I mean, the definition of that is something above normal. And so the connotation is most people, maybe non-athletes, begin to think that it's excessive. It means too high, or low is too low. I think if you step back and look at that, the origins of kind of using the word high came back on in the mid-70s with weight loss diets like the Atkins, and people were using the RDA of 0.8 grams, and then they decided to look at something higher than that, and they used double, 1.6, and they called that high. But if you look at guidelines now, like for the PROAGE study, the International Gerontology group that published a while back, they said adults should be taking 1.2 to 1.5. And the American College of Sports Nutrition recommends for athletes 1.2 to 1.8. So I don't think the word high should apply to what's being recommended as normal ranges. So I sort of put the high as something above a normal range, which is about 1.8. So that's sort of how I get to a definition. But to your point, there really isn't a definition. It's kind of like, if you're a vegetarian, you probably think 1.1 gram per kg is high. And if you're an athlete, you probably think 2 grams per kg is normal.
Yeah, exactly. And
anyway, I use a number of about 1.8. Above that, I consider that high.
Yeah. It does seem that the trend for recommending protein seems to increase more and more. Even like we're not talking about like the athletic population, we're talking about in general, especially for elderly, we're seeing higher and higher protein recommendations.
Absolutely. I think that's really been the biggest change in the protein field in the last 10 years, really. The two areas have been protein for weight loss and protein for healthy aging. And both of those show true benefits of getting up to at least 1.5 grams per kg or higher. And I think that's really changed the focus on protein, certainly in the United States in the last decade.
Yeah. Do you think the reason behind that is maybe due to limitations of measurements for protein in previous research?
I don't know that it was so much about limitations of measurements. I think we've just asked the question differently. I think that as we started looking at weight loss, we focused our attention on carbohydrates and how to control calories and thermogenesis. And we realized there was real metabolic advantages to higher protein, lower carb diets for all kinds of reasons, including lipid control. And then we started looking at things like bed rest and NASA and space flight and how to maintain body mass under catabolic conditions. And we realized that higher protein was helpful. So I think it really was a case of just asking different questions, and we realized the answers are different as we get older.
Yeah. Now, there seems to be different recommendations for younger people and elderly, but the question is there any difference in protein requirements between genders in general?
Yeah, that's interesting. I think the reality is we really haven't measured that. A lot of studies use men and women in it, and we don't really find any difference. I mean, protein is grams per kg, so when you equate a man and a woman on equal weight, it seems about the same. However, to take that a little bit further, we also probably think that protein relates to lean body mass, and women would have a little less lean body mass at equal body weight. They have a little more gender-specific body fat. But that's probably within 10% difference, and that probably is within our technical ability to measure it. For measuring differences in protein metabolism, protein synthesis, protein turnover right now, we probably need differences that approach 20% differences to detect them. And so if there's a small difference between gender at this point, it's probably outside of our detection limits. So in my experience, we find that grams per kg works for men and women equally.
Interesting. Yeah, because sometimes you hear that there should be a difference, but then again, like you said, it's based on the mass. So we're not talking about a difference in the genders per se, but it's more like because women tend to have a lower lean body mass than men.
I think theoretically it makes sense that it would be a little bit lower, but again, I think it's probably within about a 10% difference, which again is probably within our method errors at this point.
Mm-hmm. Now, there seems to be a lot of focus in the research field regarding protein on another population, which is elderly. And the question is, how should we adjust our protein requirements as we get older? And where does it where does it actually start to tip over where you should focus more on getting in extra protein?
Yeah, that's been a really hot area of research. And one of the things I would probably alert your listeners to is the use of the word requirement. One of the things that we talk about in the protein researchers talk about is differentiating sort of the concept of a minimum requirement, which is what the RDA is, versus an optimum intake. And I think that in the aging area, what we've decided is that the minimum, the RDA to prevent a deficiency, is probably about the same, maybe a little low, but in terms of optimum for healthy aging, we think it's quite a bit higher, something at least above 1.2 grams per kg. So what we've learned is that as we get older, our bodies become less efficient handling protein, maybe because of blood flow, maybe because of signaling differences, maybe because of membrane transport, but for whatever reason, we become less efficient. And we can help that efficiency with exercise, but we can also help it with just more protein. So I think in general, we have learned that a higher protein intake, better quality protein, and a better distribution of protein during the day are probably all important as we get older. And older, frankly, means end of growth. A lot of people, the research has sort of been 25-year-olds versus 65-year-olds. But from research that Doug Patton-Jones and I did, we see the transition occurring in the mid-30s. So I usually think of, quote, elderly beginning at 30.
Okay, so does that mean that you should already start to focus extra on it when you're in your 30s?
That's my opinion, is that when you get out of your 20s, when you get out of the growth period where growth hormones and insulin and IGF-1 are all really beneficial to muscle mass development, now you have to be much more careful about your diet and exercise quality because you don't have the hormones as a benefit anymore. And so distribution and things like that become much more important. And I think certainly the transition occurs by 40, but I think soon as you're ending growth, I think you need to be making that transition.
Interesting. Does it also mean that you should adjust the total amount of protein that you consume in any way, or just focus more on the distribution?
You know, I think that, you know, it depends a little on where you've decided to be when you're young. In the United States, the normal protein intake for younger adults is probably above 1.0 grams per kg anyway. The long time history is that people feel, well, when you stop growing, you need less. And that's clearly not the case. You need at least as much. If you're low, like down around 0.8 grams per kg, then you definitely need to increase it. But all of the data now suggests that for just healthy aging, you should be at least in the 1.2 to 1.5 range. So that may be an increase for some people. It may just mean maintaining what you did when you were young for others. So kind of it's an absolute target depending on, it doesn't really matter where you were before.
Yeah, exactly. And there's really no reason anyway to have a restriction in your protein intake. There's been concerns about higher protein diets having a negative effect on health, but there's been tons of research showing that for healthy individuals that doesn't seem the case.
Yeah. Bone health, kidney health, cardiovascular health, none of those have held up. There's a lot of general epidemiology out there that made some associations, but in all the controlled comparison studies, protein is always a benefit.
Exactly. And I think there was a recent meta-analysis now that came out which had 28 studies, and they basically concluded with no negative effect on renal function of any sort.
I think there were actually two meta-analyses in the last year. Stu Phillips did one, and Shailene McNeil and another group did another one. So I think there have been two on that topic in the last year.
Yeah, so there's a ton of research on it now, so there's really no concern regarding that.
It's a myth that's going to die very hard, but it is a myth.
But there's been also some speculations that having a low protein diet might actually lead to declines in renal function as well. Is there any truth to that?
Absolutely. From an exercise standpoint, it's a little like what happens to the heart with exercise. If you stress it with exercise, it gets bigger and stronger. If the kidney is confronted with more protein that it has to clear, it actually gets bigger and stronger. And the glomerular filtration rate, the rate of clearing the blood, actually becomes more efficient. when the Institute of Medicine in the United States looked at that a while back, they actually found that low protein diets were more associated with declining renal function than high protein diets.
Interesting. Yeah. All right. Now we're going to get into the real good questions. So I really want to delve into like trying to explain this so that the audience can understand it better. Could we first start by defining what the leucine threshold is, and then move on to actually to talk about its effect on activating the mTOR pathway?
Yeah. Actually, so the leucine threshold is a concept of how much leucine does it take to activate that process. But let me back up a little bit and talk to the listeners a little bit about protein turnover in different tissues. We have a tendency in muscle to think about protein turnover as a little bit of an on-off switch. But it's important to recognize that protein synthesis and breakdown is always going on, and it's very different in different tissues. For example, in the liver, in the middle of the night when you're sleeping and fasting, probably the liver rate of protein synthesis is about 70% or more of its maximum. And likewise in the heart. If protein turnover stops in tissues like that, you die. I mean, you have no leeway for it. But in skeletal muscle, which makes up nearly 50% of the body's protein, now it's more of a voluntary issue, and it seems to be more controlled by meals. And so it will run along at more like 30% of maximum in a fasted condition, and then after a meal, it will go up by 2 or 3-fold. And so what we have is a muscle is a tissue that is very meal sensitive, especially in adults, versus liver, which is not very meal sensitive. And if you look at how the body regulates that, in the liver, the protein synthesis is regulated by a series of initiation factors, one of which is called initiation factor 2, EIF2, and that one's sensitive to energy. Where in muscle, it is EIF2 is generally not limiting, and it's regulated more by EIF4, which is very sensitive to protein. Okay, so that brings us back to the difference with mTOR. mTOR is a sort of a master control system that regulates protein synthesis at the initiation phase. And it's sensitive to at least 4 different things. It's sensitive to the amount of energy around, which is sensed by AMP kinase. It's sensitive to energy because of insulin, hormones, IGF-1. And it's sensitive to protein in the form of leucine, and it's sensitive to other things such as exercise. And the muscle integrates all of these for how it's going to respond. And so for whatever reasons, and we don't really know why, the body evolved to sense leucine. And interestingly enough, the body also evolved not to metabolize branched-chain amino acids in the liver. So unlike all the other amino acids, after you eat a meal of protein, the amino acid leucine goes almost directly into the blood, really not altered much by the liver, and then goes out to skeletal muscle, which then senses that meal. And again, for reasons we don't really know, what was the teleological reasoning behind it, but the body learned to sense an amount of leucine. And it turns out that it takes about 2.5 grams of leucine to turn on mTOR in muscle. When we're young, mTOR is turned on mostly by insulin and growth factors, which is why we were talking about aging. When we're young, we're not nearly as sensitive to protein at meals. As we get older and stop growing, we're not as sensitive to insulin, we're not as sensitive to IGF-1, and now we become more sensitive to the amount of protein. And it turns out it takes about 2.5 grams of leucine to actually stimulate mTOR. And so you'll see numbers like 2.5 or 3.0, and that's kind of a threshold. If you're below that, if you're at 1.5 grams of leucine, it won't turn on mTOR in muscle. It will turn it on in liver because liver is more sensitive to energy, but it won't turn it on in muscle. So the aspect that muscle becomes sensitive to this leucine threshold, I'm probably guilty of sort of coining the concept of it being a threshold. We know that 1.5 grams doesn't work and that 2.5 grams does work. We don't really know what it looks like between 1.5 and 2.5. It's kind of, we treat it like an on-off switch. We don't really know. Our techniques aren't such that we can tell the difference, what's the shape of that curve. So, but it's kind of a threshold. You've got to get above 2.5 before you really get much of an effect.
Interesting. And we're talking about, in these terms, we're talking about absolute amounts, or do we also have any guidelines for based on body weight or something like that for how much you need?
It's interesting. Almost all the data has shown, regardless of body weight, that 2.5 to 3.0 works. Leucine's kind of unusual in that it probably doesn't relate to lean body mass as much as it relates to blood volume. Mm-hmm. And the blood volume in a person who's 150 pounds versus 250 pounds isn't very different. 4.5 liters to maybe 4.8. So they might be vastly different in weight, but the blood volume didn't change that much. And really what it takes for leucine to work is the blood level has to go up by about threefold from baseline. Mm-hmm. The other thing, sort of thinking about this threshold, is that, and the whole initiation process, is that it's the first step in protein synthesis. And I think of leucine as really a recovery from a catabolic condition. In the sports world, people have sort of tried to think about it as like an anabolic hormone, that you can take a bunch of leucine and stimulate muscle mass. That's not really what it does. What it does is recover from a catabolic condition. Like when you are fasting overnight for 12 hours and you wake up in the morning, your muscle protein synthesis has been shut down. And to re-trigger that, you have to trigger mTOR. Mm-hmm. What mTOR does, I mentioned earlier that there's sort of this baseline protein synthesis, and in muscle it's like 30% active even when you're fasting. And so what you're trying to do is stimulate it. What mTOR does is actually target certain mRNAs that increase capacity. And so if you look really closely in the literature, you'll see it has somewhat more effect on myofibrillar protein than sarcoplasmic protein. And because it's turning on certain messenger RNAs to increase your ribosome number and things like that. So it's targeting very specific mRNAs, and it's starting the process. And so, giving more leucine, once you have enough, giving twice as much won't do any more. If the meal has 2.5 grams of leucine in it, giving 2 more grams won't do any more. You've already started the process, and you can't, it's like turning on the car engine, turning the key on the car. Once it's running, you can't turn the key on again and get any more. Just once it's running, it's going.
Yeah, exactly. And this has had this, especially in the supplement industry, it's been a huge hype with supplementing more leucine in the diet, because the question has been amongst a lot of people in the fitness industry, if leucine is most important to activate this pathway, why do I need to eat the other stuff? Why can't I just take leucine and be done with it?
Sure. The important thing there is leucine just simply turns on the machinery. Mm-hmm. And once the machinery's turned on, then you need all of the parts to put together the car. Mm-hmm. So if you don't have all the other amino acids, it's not going to go anywhere. And so once leucine turns on mTOR and activates these mRNAs, now you're responsible for all of protein synthesis. And what we have shown and others have shown is that if you just give leucine, you'll start dropping the blood levels of all the other amino acids. You'll suck them out of the blood. And so you might get a momentary burst of protein synthesis, but it won't last very long because you run out of substrates.
Interesting. So it's basically like trying to build a building where you have workers and building material. So if you don't have the building material, you won't be able to build anything.
Exactly. You can tell them all to get started, but if they don't have anything to put together, it goes nowhere.
Yeah. But we mentioned the amount of leucine that you need for each meal, but it's not really a practical recommendation maybe to tell people that you need this amount of leucine to each meal. So do we have anything that's a bit more practical to recommend with examples of amounts of protein you need for each meal, for example?
The 2.5 grams was actually a number we coined because in the United States, the protein mix between animal protein and plant protein is about 70-30. We get about 70% of our protein from animal, 30% from plants. And if you look at that in a mixed meal, that mixed meal will have about 8% leucine. And so if you'd use that number of 8%, 30 grams of protein will give you about 2.5 grams of leucine. So 30 grams of protein became kind of the target. If your meal is all whey protein, for example, whey protein is around 11% leucine. So you could get away with about 22 grams of protein and stimulate muscle protein synthesis, which is great for things like after exercise or small meals. But if you're a vegetarian, you want to use just plant proteins, you might have to have 35 to 40 grams to get to that level of leucine. So, we use a mixed meal concept of about 30 grams, but again, it depends on the quality of the protein you're selecting.
Excellent. Now, the follow-up question to that would be, if more doesn't necessarily give you a better effect, what actually happens to the excess protein that you would consume in a meal? Let's say if 30 grams is what you need to hit that leucine threshold, what actually happens to the extra amino acids that you've consumed?
So that's a really good question. And the way you ask it, it's important because it's the way a lot of people will think about it. Again, leucine is a threshold for turning on the system. That doesn't mean the maximum amount of amino acids you can get a benefit from. So having more protein will actually increase your body's ability to make protein. So that we're separating the leucine question from the total protein question. Okay, so is a 50-gram protein meal better than a 30-gram protein meal? And I would say that if you're a bodybuilder, yes, it is. Okay, so 30 gram would be the minimum to get a muscle effect. So if you're doing weight loss, for example, and you're trying to maximize your muscle benefit with the least calories, then 30 is a good target. If you're trying to build muscle mass, then you should probably have 1 or 2 meals per day that hit 50 grams because you're trying to hypertrophy. So we need to separate the leucine concept of the starter mechanism from the maximum effect of amino acids. Your concept of where do the extra amino acids go, for adults who are not changing body mass, every amino acid that you eat per day is oxidized, whether you eat 100 grams or 200 grams. That's just the physics of it. Your body is a constant amount of protein, so that means if you take in 100 grams per day, you have to oxidize all 100. Mm-hmm. If you take in 200, you have to oxidize all 200. So there's this concept of inefficient use, but the reality is there's not a really direct correlation to efficiency of use and protein amount anyway. That's really a misnomer in the literature.
Yeah, because sometimes you'll hear, especially in the fitness industry, you'll hear people say, well, if you consume more than 30 grams of protein, you'll just pee out the excess protein that you've consumed, which isn't the case.
I mean, no matter what amount of protein you consume, you will pee out the nitrogen. So whether it's 40 grams or 100 grams or 150, it all will end up as nitrogen in urine. So,
yeah.
The issue is efficiency again. So, you probably get your maximum effect for the least protein at around 30. But you'll hear trainers say, well, you can't use more than 30 at a time, and that's totally not true. You can use 40, you can use 50. And in fact, when I make recommendations to people, I make recommendations of uneven meals. I would like to see them have a couple of meals that are one's 30, one's 40, one's 55 or 60. I like having different amounts of meals because the body reacts to them differently.
Interesting. So would that mean that you would have some feedings that would be pretty high in protein content and then some smaller meals throughout the day?
Exactly. And I think that for most of our lifestyles, certain meals are easier to get higher protein into. In an American lifestyle, typically breakfast, the first meal is fairly light and the last meal is fairly heavy. I think that's perfectly fine. If you want to reverse them, that's perfectly fine. But I like the body adjusting to different meal size. There's sort of the concept that came out of the leucine that we should have an even distribution of protein. And I don't think the literature supports that at all. I think having a different distribution where some meals are higher is actually much better.
Okay, interesting. As long as you are hitting the threshold for each meal.
The threshold.
Yeah.
And frankly, when I look at it, I think the first meal and the last meal are probably the more important ones, and we can go into that in more detail. But the one that when you wake up after an overnight sleep is probably the single most important one in terms of turning on the machinery, because it's in a catabolic state, and until you get that leucine threshold exceeded, you're going to stay in a catabolic state breaking down protein. So that first meal, and I usually call it the first meal versus breakfast because I don't care whether it happens at 7 in the morning or noon. The first time you eat, you need to get it right.
Yeah, exactly. Yeah, there's been a ton of research on especially the meal pre-bed, especially coming out from Van Loon's lab in the Netherlands. And it does seem to suggest maybe that it's beneficial to at least make sure that you're consuming adequate amounts or higher amounts of protein before going to bed.
Again, I think if you're really looking to be a bodybuilder, I think looking at 4 meals, 4 different distributions, one of which is just before bed, is again really good. Again, if your goal is healthy aging, body weight control, adding a 4th meal in probably is counterproductive. So again, I think if you're trying to maximize muscle mass, it's a good idea. I don't think the benefit outweighs the calories for most people.
Yeah. One thing that I also wanted to mention, that you were saying that 30 seems to be the minimum. If you actually look at the protein dosage studies that we have the study by Moore from 2009, we have a study by Wittard from 2014, and then we have a McNaughton study that came out in 2016, I think, where they're comparing different amounts. If people would look at the graphs, because 2 of the studies conclude with 20 grams being the maximum amount you need to maximize muscle protein synthesis. But if you look at the graph, it actually is increasing when you're going from 20 to 40. It's just that the difference isn't significant enough for them to conclude that. But the difference was the McNaughton study, which actually showed 40 grams was better than 20 grams. And what they concluded with was it probably has something to do with more muscles being trained compared to single muscle groups being trained. So if you were to do a whole body routine, it would require more proteins in a meal.
The two things to be very sensitive to when you're looking at those papers is, one is what it was the age of the subjects, and the other is, are you talking about post-exercise or not? Exercise lowers the threshold. So when we talk about 30 grams, that would be like a morning breakfast without exercise. But if you have an exercise, a resistance type exercise preceding it, you'll probably get an effect with as little as 15 grams. 15, 20 grams will probably be okay. The more paper, if I remember correctly, was young adults. I think that was 25-year-olds. And so again, they're much more sensitive to protein than older adults. And I think that the other one that out of the Van Loon's lab was older adults. I believe that was more 60-year-old type individuals, and they found the more 40-gram target. So, I think you need to be, are you talking about post-exercise? That's different than just a normal meal effect, because the exercise increases the efficiency of use, maybe because of blood flow, maybe because of signaling of mTOR, both of which we know to be true. And so it increases the efficiency. And then aging decreases the efficiency. So those are the two things to keep in mind as you look at the literature and try and sort through the noise of different conclusions.
Interesting. Now, moving on to, you briefly mentioned the AMPK, which I wanted to talk more about. So let's start off by mentioning what is AMPK and what effect does it have on muscle protein synthesis?
AMPK is a really cool enzyme complex, and it's often referred to as a master energy regulator of the cell. So we will trigger AMPK, AMP kinase, in conditions of low ATP. And so so the ratio of AMP to ATP will increase, and that's what triggers it. And so the body will then begin to do things to either generate ATP or conserve ATP. And so notably, it increases things like glucose transport into cells. It'll increase GLUT4 transporter. It shuts down fatty acid synthesis and increases fatty acid oxidation, things that increase AMP or ATP. In terms of protein synthesis is an amazingly energy expensive process. And so AMP kinase will shut it down. It inhibits mTOR, which sort of slowing down initiation. So when you wake up after an overnight fast, AMP kinase would be activated, and that's shutting down mTOR, and you're in a catabolic condition. AMP kinase is also activated, say, after a meal, and it shuts it down in muscle because we're burning up ATP, and it will then inhibit the elongation factor 2. So if you look at protein synthesis, first we initiate the process, which just sort of gets all the machinery together, and then we have the elongation process of actually making the protein, putting each individual amino acid together, and that's actually where all the energy is. And so AMP kinase directly inhibits elongation factor 2, EEF2. It slows it down and conserves ATP in the cell. And we actually showed that. Gabe Wilson, Lane Norton, in my lab showed that after a meal, protein synthesis will slow down, and it's primarily because of activation of AMP kinase.
Interesting. Now, is there anything other than, like you mentioned, when you wake up in the morning after a fast, AMPK will be activated? What effect does exercise have on AMPK?
AMPK will also be activated with exercise because it's an ATP expenditure. And so it will trigger all of those processes, increase glucose transport, increase gluconeogenesis, increase fatty acid synthesis, and slowing down protein synthesis. So exercise itself is a catabolic condition. We're burning fat, we're slowing down protein synthesis, et cetera, et cetera. So AMP kinase is very important in exercise, starvation, fasting, any catabolic condition.
Interesting. So would that mean that it would be activated in the post-exercise window where people are recommended to consume nutrients? And is it only...
So it's activated during the exercise, and then in that post-exercise recovery, that's why you're taking in the nutrients. You're trying to counterbalance that stress. And AMP kinase is one of the things you're trying to reverse.
And is it only protein that you should be focusing on? Is it only protein that has an effect on that? Or does carbohydrates also affect it?
I haven't studied that as well, but glucose will also help reduce the AMP kinase. And you'll see some people, and we've done some studies giving just protein or just glucose or a combination. And so I think there's benefit to having both. Again, people have sort of studied it relative to glycogen repletion as opposed to ATP or AMP kinase effects. But the effects are on both. When you are in an energy limiting situation, the body will use protein as an energy source too. Branched chain amino acids are a very effective energy source. So I see benefit to actually using protein and some carbs, especially if you're going to perform again soon. If you're really just looking at bodybuilding, probably less important. But if you're an athlete who's going to perform again, say a tennis player or something, taking in both probably makes some sense.
Excellent. Now you mentioned BCAAs, and that seems to be a really hot supplement in the industries. So what role does BCAAs have on muscle protein synthesis, and do you think it's beneficial to supplement BCAAs in your diet, or are we getting sufficient amounts from the protein sources we're eating?
In general, we're getting sufficient amounts, but back up one step. So BCAAs, branched chain amino acids, are leucine, valine, and isoleucine, the three branched chain. And so we've talked about leucine. Is it useful to supplement leucine? The question there would be, can you reach your leucine threshold with your protein? For example, a lot of people at lunch might go out and have a salad that maybe only has 15 grams of protein in it. Taking in a branched chain amino acid supplement at that point would help you reach the 2.5 grams of leucine and trigger protein synthesis, even though you only had 15 grams of protein there. So for vegetarians, for small meals, for breakfast, for places where you're less than 30 grams of protein, it might be useful. If you've got 30 grams of protein, so you've reached your leucine threshold, then there's no benefit at all. They really just become an extra amino acid source. I think there's room for them, but only in cases where protein is inherently low. If you're going to take a supplement, should you take leucine or branched chain amino acid, all three of them? What we know is that when you trigger protein synthesis, we could talk about TCA cycle things and why exactly, but when you trigger protein synthesis, you also trigger leucine oxidation. And when you trigger leucine oxidation, you begin to deplete the TCA cycle. And so for every molecule of leucine metabolized, you're removing an alpha-ketoglutarate. And to replace that, you need to have a valine or an isoleucine, which will replace it sort of at the next step, succinyl-CoA. And so basically, you need to take all three branched chain amino acids at approximately a 2-to-1-to-1 ratio. So when you see supplements, you'll typically see them in that ratio. And that's because that's how they're metabolized. And if you look in the blood, if you just give leucine alone, I mentioned before that you'll start depleting all of the amino acids in the blood because you're triggered protein synthesis. Well, you'll deplete valine and isoleucine twice as fast as any others because of the metabolism I just gave you. So if you're going to take a leucine supplement, you really should take a branched chain amino acid supplement in about 2-to-1-to-1.
Interestingly. Yeah, because now in the industry, you've seen that there's been, ever since leucine got all of this attention, a lot of the supplement companies, they started making different formulas for BCAAs, you would see the 4:1:1 ratio, 8:1:1 ratio, which may not be as beneficial as people might think.
Yeah, I think the ideal ratio is about a 2:1:1. And again, as a supplement, it's really only beneficial with a lower protein diet. There have been a few studies that use branched chain amino acid during endurance exercise, and about 50% of them showed some increase in endurance, kind of like taking, a carbohydrate supplement. But I think most people would agree that carbohydrate supplements are more effective energy sources during an exercise than a branched chain amino acid. But they are good energy sources, and they are the direct precursors to alanine production and also glutamine production, which are two really important fuels. Alanine is very important for recycling glucose during endurance exercise. There's some evidence that during prolonged cycling, that 40% of the blood glucose can be from gluconeogenesis from alanine. And to fuel that, you have to have branched chain amino acids because they generate the nitrogen that you use pyruvate. So you use glucose in the muscle to get pyruvate, glycolysis. It traps the pyruvate as alanine, sends it back to the liver, and you reuse it all over again. So you have this great potential to recycle glucose during endurance exercise.
Interesting. Moving on to the next question. We sometimes hear the term refractory periods when we're talking about protein. Could you explain what that means?
Yeah, that's really a cool area. Phil Atherton and Mike Rennie and my group have sort of gotten into looking at that. One of the things we know is we've focused on protein synthesis after a meal, and it triggers it, and it goes up really rapidly, and we know leucine's a trigger. And so we started asking, well, how long does that run? And what we find is that after a meal, it runs for about 2 hours, and it will come back to baseline. And it doesn't seem to matter how much is in the meal. It doesn't really matter too much about it. It runs for a period and comes back down. And when you start looking at all the mechanisms, the amino acids are still high in the blood, leucine's still high, mTOR is still activated, the initiation factors are still activated. And so why does it come back down? And so I think Phil Atherton coined the concept of muscle full, whatever. I like the term refractory better in that basically the muscle just isn't able to continue this expensive process. We actually looked at that, and what we found was ATP is actually getting depleted in muscle cell. And you can kind of imagine that the last thing you'd want for survival is to have your muscles unable to function. You're out in the wild running from your last kill or something of a wild animal, and you can't move. So basically, the body has a way of protecting ATP levels, and in fact, it triggers AMP kinase. Mm-hmm. And so we think, my lab paper we published, American Journal of Physiology, is we think it's an AMP kinase mechanism that is slowing down the elongation factor 2. And basically, the muscle is refractory to any sort of input until you get ATP built back up. We think that period is around 5 hours. Mm-hmm. And so sometimes we will talk about meal distribution of around 5 hours. That if you take in another protein meal within 3 hours after a protein meal, there's a good chance it'll have very little effect because you're in this refractory period. And so we like a spacing of 4 to 5 hours between meal so the muscle has the time to reset and be ready to actually efficiently handle the next meal.
Interesting. So in practice, you would have longer periods between each meal.
One of the reasons I mentioned earlier that I like the first meal and the last meal is because you now have a lot of distance. You're going to get the maximum effect of the protein you eat because the system has totally reset.
Interesting. Now, I don't remember the author. I think maybe it was Gabe Wilson that did a paper on this, but if I remember correctly, he looked at supplementing branched-chain amino acids between meals to see if it had an effect on the refractory period.
Exactly. We did that. So Gabe and Lane Norton in my lab. So we studied the effect of that refractory period, and we realized it was AMP kinase and the elongation factor 2. So then we started asking, well, if it's an ATP, can we supplement energy? So we started giving different glucose combinations or leucine combinations, and we found that we could actually extend, if we gave the supplement at the top, kind of at the peak of the protein synthesis, that we could actually extend the period of protein synthesis. We did it twice. It worked in our laboratory. I know a couple of other laboratories that have tried to repeat that. I think Phil Etherton tried, and Dominic Dardavat tried, and I don't think either one of them found that. So at this point, I'm reasonably comfortable with the fact that AMP kinase is involved in the refractory issue. It relates to ATP and the elongation factor 2. Do supplements really mitigate that? I think that's still a question up for grabs. You know, we found it, we repeated it, but other labs haven't. So I don't know if that's the conditions we used versus theirs. I think it's an open question, and you know, I still don't know the answer to it. I still think for efficiency, thinking about 4 to 5 hours between meals is the right thing, and supplementing in between, I think, has little effect. So I think that's where I would sit at this point.
Okay, interestingly. So can I ask, what time did you actually incorporate the supplementation? Like
So in our model, which was actually, we were using rodents, we were using rats, the period of protein synthesis was, if I remember correctly, about 180 minutes, and it came back to baseline. And so we gave the supplement, I think, right around 100 minutes. So we were right after the peak. So protein synthesis peaked between 60 and 90. It was sort of at a peak. And so we got it just as it was beginning to fall out of the peak. So it was still, everything was active, everything was maxed. Protein, you know, AMP or ATP was still high, and that's where we gave the supplement. So again, did we just happen to hit it coincidentally? I don't know. You know, it's still an experimental question. But what we do know is that there is this refractory period. Protein synthesis comes down when all of the indicators are it still should be running. You know, leucine's high, mTOR's on, all of the initiation factors are triggered, but yet it stops. And so, again, we think it's an energy issue, an AMP kinase. Can you manipulate that? Open question.
Interesting. All right, this was great. I think we covered a lot of things in this podcast, but before we sign off, could you please give the listeners a take-home message to conclude this podcast?
Yeah, I think I would probably go that, you know, athletes recognize the importance of muscle, but I think we're recognizing it more and more just for general health, that muscle is really a key to fitness and long-term health. I like the concept of a muscle-centric view of health. And I think that what we've recognized now is that protein and exercise, particularly resistance exercise, are important for everyone. And that as we get a little older, the amount of protein at each meal, the distribution, the quality of protein all become very important. And we used to think it was only about children and growing, but it's really a lifelong issue now that we maintain our muscle health and we pay attention to the combination of dietary protein and exercise.
Excellent. All right, Professor Layman, thank you so much for taking the time to be on the podcast today. For the listeners out there, is there anywhere they can find more information about you? Are you active on any social media platforms?
I do participate in Twitter, so my handle is at Don Layman. So I basically tweet about science, so that's a good place to follow. And you can certainly find me. My email is dlayman at illinois.edu, so you can track me down. And I'm pretty widely published, so you can find me on Google.
Excellent. Once again, Professor Layman, thank you so much for your time. I hope you enjoy the rest of the day, and hopefully it doesn't get too cold in Chicago today.
Winter is arriving, but thank you. It was my pleasure to join you. Thanks.
Thank you so much. Bye-bye.